The concept that all species are related by common ancestry and have changed over time in response to natural selection is the foundation for understanding the tree of life. The tree of life depicts the concept that evolutionary history of species traces back to a common ancestor. The tree of life is a family tree representing the relatedness or phylogeny of all living groups of organisms. By analyzing genetic variations of species, a phylogenetic tree can be produced to arrange branches to align with genetic data. The tree of life is still a work in progress, but let's explore how these trees attempt to describe the history of life, applying concepts of evolution. Populations don't only develop just new traits over time, they can do more than that. Natural selection has caused populations of one species to diverge and form entirely new species. This divergence process is called speciation, and by applying evolution and cell theories, scientists predict that all species come from pre-existing species with traceable ancestry back to a single common ancestor. The theory of evolution by natural selection predicts the possibility to construct a tree of life, the family tree of all organisms. If life arose once, the diagram would describe the relationships between species with a single ancestral species at its base. A breakthrough in research on the tree of life occurred when biologist Carl Woese and his colleagues began analyzing the chemical components of organisms as a way to understand their evolutionary relationships. To determine the phylogeny of all organisms, now translated literally, phylogeny means tribe source. To discern how closely different species are related, Woese studied a molecule that is found in all organisms. Specifically, they selected small subunit ribosomal RNA, since it is an ancient and also found in all known life forms on Earth. It is the smaller of the two major RNA components of the ribosome, and generally, ribosomal RNA is a large and complex molecule, but its underlying structure is simple. The ribosomal RNA molecule is made up of sequences of 4 smaller chemical components called ribonucleotides. These ribonucleotides are symbolized by the letters A, U, C, and G. In ribosomal RNA, ribonucleotides are connected to one another linearly like the boxcars of a freight train. Why do you think might be the reason ribosomal RNA can be useful for understanding the relationships between organisms? Well, the answer is that the ribonucleotide sequence in ribosomal RNA can change during the course of evolution. Although ribosomal RNA performs the same function in all organisms, the sequence of the ribonucleotide building blocks in this molecule is not identical among species. Woese and his colleagues pursued their research with a simple premise: that ribosomal RNA sequences should be more similar in closely related organisms and less similar in organisms that are less closely related. Furthermore, the species that are part of the same evolutionary lineage, like the plants, should share certain changes in ribosomal RNA that no other species have. And it turns out that they do. A diagram that depicts evolutionary history in this way is called a phylogenetic tree. Just as a family tree shows relationships between individuals, a phylogenetic tree shows relationships between species. On a phylogenetic tree, branches that share a recent common ancestor represent species that are closely related. Branches that don't share recent common ancestors represent species that are more distantly related. The tree of life, implied by ribosomal RNA, established that there are 3 fundamental groups or lineages of organisms: bacteria, archaea, and the eukarya. All eukaryote cells have a prominent component called the nucleus. Translated literally, the word eukaryotes means true kernel. The vast majority of bacterial and archaeal cells lack a nucleus, though. They are referred to as prokaryotes, meaning before kernel. Bacteria and archaea are typically unicellular, while many eukaryotes are multicellular. We can differentiate eukaryotic cells from prokaryotic cells, since prokaryotic cells do not have the membrane-bound nucleus. When results based on genetic data were first published, biologists were very surprised. Prior to the tree of life, the division among organisms was thought to be between prokaryotes and eukaryotes. But the archaea were virtually unknown, nor were they even recognized as a major branch on the tree of life. Fungi were thought to be closely related to plants, but instead they are actually much more closely related to animals. Traditional approaches for classifying organisms include the system of 5 kingdoms. They're divided into various classes, orders, and families that you may have learned in high school and are actually inaccurate. That's because they don't really reflect the evolutionary history of the organisms involved.